Perfect. Okay. Hi, everybody, and welcome to today's webinar. This is the Plant Physiology webinar focusing on plant lipids, and it's in celebration of the February 2025 focus issue on plant lipids, metabolism, and signaling. Today, we're joined by three speakers whose work appears in this focus issue, Rebecca, Yingqi Cai, and Philip. We have the host, Aruna Laxalt, and our moderator is an assistant features editor, Maneesh Lingwan, who will be introducing the speakers and also fielding the questions from the audience. The focus issue is online now. Not all of the articles that will be in it have appeared quite yet, but most of them are online and they will be appearing in the next few days. So please visit Plant Physiology to have a look at some of these great articles. The editors are Aruna Kilaru, Aruna Laxalt, Sébastien Mongrand, and Teun Munnik, and we thank them for their work in putting together this very impressive focus issue. We will be recording this and posting it to YouTube. And if you visit our Plant YouTube channel, you'll find all of the focus issue webinars for plant physiology and plant cell. There's a lot of great science there. We want to make a special thank you to ASPB members whose membership dues support the running of these webinar series. If you'd like to join ASPB, you can use the code PRESENTS10 and you'll get 10% off your membership dues. We would also like to invite you to join us this July in Milwaukee, Wisconsin for the annual plant biology meeting. It's a great meeting. I've heard Milwaukee is lovely, although I've never been myself, but I'll be there. And I'd like to remind you also that if you register by March 19th, you'll get a discounted registration rate compared to later registrants. If you have questions for the speakers, please put them in the Q&A box and Maneesh will answer some of them. You can email me if you have any questions. If you need a certificate of attendance, all attendees, live attendees, will receive an email tomorrow with instructions on how to get that. And so now I'm going to turn it over to Maneesh, but first of all, I will introduce Maneesh, tell you who this is. Well, as I said, Maneesh is an assistant features editor at Plant Physiology. He's also a research scientist one at the Donald Danforth Plant Science Center in St. Louis, Missouri. He's a passionate plant biochemist. He's interested in analytical techniques and strategies to decode cell-specific metabolism. He's currently researching plant lipid metabolism to enhance medium-chain fatty acids in oil crops. But besides research, Maneesh he tries to inspire and educate younger students by providing valuable insights about STEM careers and to help them navigate opportunities. So thank you, Maneesh, for hosting, and over to you.
Thank you, Mary. First of all, I would like to welcome our speakers and everyone joining us today for the webinar. Our webinar host is Aruna Laxalt. She is a professor at Institute of Biological Science, National University at Mar del Plata, Argentina. Her lab studies the role of lipid signaling in plant stress with a special focus on plant defense responses. She studies the role of different phospholipases, C and D, in the perception of pathogens and the induction of defense responses, including stomata movement. She is interested in establishing the role of lipid second messengers in relation to the other second messengers involved in a plant defense, such as reactive oxygen species, calcium, and the gastrotransmitters like nitric oxides and hydrogen sulfides. Over to you, Aruna.
Thank you. Thank you very much. I will share the slides. So, good morning and good afternoon and good evening, depending on where you are. Thanks for the presentation. I'm Aruna Laxalt, monitor editor for Plant Phys and a professor at the University of Mar del Plata. It is an honor to welcome you to this webinar, which celebrates this February issue of 2025 focus issue on plant lipids, signaling and metabolisms. Plant lipids play vital roles not only as structural components of the membranes, but also as signaling molecules and regulators of metabolic pathways. This focus issue, as Mary introduced, was edited by Aruna Kilaru, Sébastien Mongrand, Teun Munnik, and myself, and gathers groundbreaking research articles, reviews, and updates to explore the multifaceted roles of lipids in plant growth, development, and responses to environmental cues. So this focus issue aims to prove a comprehensive overview of the role of lipids in signaling and metabolisms. And we launched like this invitation for a focus issue like 1.5 years ago, and researchers were invited. And today, this is how the cover looks like of the focus issue. This is the Plant Physiology cover. It's signaling and metabolism. The volume is in 187. It's in February. It is still not complete. It's in progress if you look at in the page. And today we are privileged to have 3 outstanding speakers who have contributed significantly to this issue. And that's why we invited them to share their seminar with us. They will share cutting-edge research spanning topics such as membrane lipid remodeling under stress, lipid droplet biogenesis, and lipid metabolism dynamics in plants. Their work exemplifies the depth of their research in plant lipid biology. This cover is bodied by stained lipid droplets in castor seeds, the source of valuable castor oil. So we have like 1 topical review, 1 research report, 11 updates, which is a lot, but the most astounding is that we have 17 research articles. And if we divided them like in 5 different topics, and I will go through like very rapidly. So we have some inputs on transcriptional and genetic regulation of lipid metabolism that covers transcription factors regulating lipid accumulation and genetic modification influencing biosynthesis. So the other one is biosynthesis and metabolisms of plant lipids, the regulation of very long chain fatty acids and specific lipid biosynthesis, mechanism of controlling sphingolipids and triacylglycerol homeostasis, the role of membrane structure in lipid stability and function, and the formation of and function of lipid droplets in plants. And then we have lipid signaling, stress responses, and development. So membrane lipid remodeling in responses to environmental stress, phosphonatidides, phospholipases, phosphatases, and kinases, and the role on cellular signaling networks during different stresses and development. And the involvement of lipids in plant defense against pathogens and also volatiles are there. So, and then we have lipid interactions with the cellular, the relationship between lipids and autophagy in plant cells, and regulation of lipid metabolisms by TOR kinases. And we end up with a biotechnological applications and crop improvements, the use of Camelina sativa in lipid biotechnology, strategies to improve oil quality and compositions in seeds, modifying lipid metabolisms to enhance stress tolerance in crops, and multi-omics analysis applied to crops for improving biofuel and vegetable oil production. So with having said that, I want to acknowledge to Maneesh, the assistant feature editor that is handling the webinar, to the speakers, to the scientists that contribute in this focus issue, to all the papers that we have in the focus issue, to all the participants that they are together and joined today to this webinar, which is what makes the field going forward and very exciting, and maybe some collaborations we will have end up after this webinar. And to Mary for organizing the webinar, of course. And I give the word, and thanks again.
Thank you, Anna, for compiling all the excellent research and briefing for us. Now it's my pleasure to introduce our first speaker, Rebecca Roston. She is an associate professor in the Department of Biochemistry at the University of Nebraska-Lincoln. Her research focuses on the molecular and the biophysical mechanism underpinning the plant membrane adaptations to environmental stresses, particularly cold and freezing conditions. Her lab investigates the dynamic processes of membrane, lipid remodeling, and the protein interactions and their roles in the plant growth and photosynthetic efficiencies. She uses advanced microscopy, molecular biology, and biochemical techniques. Her work spans model systems like Arabidopsis thaliana and diverse grasses to uncover, conserve stress responses, and to improve the plant resilience. Beyond research, she is committed to education and mentorship, teaching and advising the students and postdoc organizations on CROPS, CROPS. Today, she will tell us about the role of membrane lipid remodeling in plant responses to low temperatures. Over to you, Rebecca, and thank you.
Thank you for the very kind introduction. And also, I want to say a big thanks to Anna, Aruna, Sébastien, and Teun, because it is a lot of effort to do one of these special issues. And I super appreciate your service to our community by organizing this very, very nice work. So thank you. And of course, thank you for allowing me to speak to you today. So as Maneesh told you, I'm going to talk about low temperatures. What am I going to tell you about low temperature? I'm going to tell you about this update article that was written by Kaylin, Zach, and Fang Yi, as well as a high school researcher, Sydney, who wrote 4 pages of introduction, which ended up being deeply cut, but we appreciated her efforts. First, I'm going to tell you about why I find cold interesting. And then I'm going to tell you about the really beautiful things that happen to lipids during cold. Some of our current knowledge and updates, a brief summary of efforts from our lab, because, well, I would be remiss not to take the opportunity, and then some open questions, including those from ours. Start really basic here. This is the average calories eaten per day per person in the world, and we eat a lot of grains. Maybe we could argue about too much or too little of other things, but for grains specifically, cold is a huge issue. This is a heat map, essentially, of a world map in which grain production is darker when there's more of it. And what you can see is that compared to the lovely, here we are, the lovely equator, these are all happening in temperate regions. So that means that right now in Nebraska, where I am, the fields look like this. And I think you can appreciate the sincere lack of productivity in that field at the moment. And why is that? That's because we're human. We came from warm areas of the world, and crops were domesticated in these warm areas of the world, and we brought them with us because we liked them. But now we're facing this problem where food is not as abundant as it used to be. And when we compare crops like maize to more cold tolerant crops that could grow earlier and later in the season, it becomes obvious that cold is limiting our agricultural productivity. So that's the reason why I think cold is important, because I like eating. Now let's talk about the really cool stuff, how cold affects membranes. So you're probably already familiar with some of the effects of temperature on membranes from your kitchen. You may have cooked with olive oil, which is a liquid at room temperature, and stuck it in the refrigerator, solidifying it. You may have also set butter out in the summer and liquefied it. And this is just one indicator that the biological lipids are, in fact, going through relevant temperature phase changes. In membranes, that looks a little bit more complex, but it means some really obvious things. So at high temperatures, lipids are not as ordered, and that means membranes leak. And this is very clearly measured by a large number of people. This is just one of many examples. As they get colder, leakage stops, but this isn't a perfect system that is not disrupted. What actually happens is that phase separations occur where gel phases are formed. These crack out of membranes, causing fractures that, especially when cold temperatures thaw, are huge problems in any system. And then plants have this whole specific thing that happens. So this is a thing that is not normal in animal systems in cold, but in plants it's observed directly, and that's hexagonal two-phase fusions formed between different membranes, possibly because plants have so many more non-bilayer forming lipids that when separated out and in close apposition tend to form these fusions. Okay. So how do we fix it, and how do plants fix it? Part of that answer is by changing the lipids. Let's talk about where the lipids are first. Plant membrane lipids are actually a complex network. I've heard it called a superhighway, I think by Phil, who will talk to you later. But until then, you'll have to deal with my lipid network here. They start getting made in the chloroplast with free fatty acids, where they're connected onto lipid head groups and then progressively get more desaturated by desaturases in each of those compartments. They're also trafficked to other places. So through the secretory pathway, they're trafficked to the plasma membrane. The plasma membrane is super specific. In fact, there's an entirely different class of lipids there. So far, we've been talking about lipids that have a glycerol backbone, but there are also lipids that have a long chain base background. And so these are ceramides and sphingolipids, and they form the very important surface of the plasma membrane, which is the connection between the cell and the outer world. In addition, there are specific lipids formed in both the mitochondria and the chloroplast that we believe are essential for their function because they're conserved throughout life. So the secretory pathway is actually not enough, although of course that helps form the plasma membrane, vacuole, Golgi, lysosome, and endosomal compartments. Redistribution of lipids happens very rapidly through membrane contact sites. And we know now from the mammalian field that these are ubiquitous. We're just discovering how ubiquitous they are in the plant world now. So we believe these are part of how some of the lipid redistributions that are observed between normal and stress conditions. So this is the normal cell, and this is your cell on cold, or your plant cell on cold. And you can see there's some pretty dramatic changes. Some of the first are actually that the plasma membrane starts stiffening because of the cold. This activates some calcium channels over here, and that sets off a bunch of transcriptional responses, which I won't talk about because this is not that type of seminar. This is about the lipids. So in addition to this signal, which we know happens very quickly, there's another set of signals that we know is happening very quickly. There's some phospholipases here that are making a large amount of phosphatidic acid at the plasma membrane, which may actually be the junction between the plasma membrane and the endoplasmic reticulum. We know that these are trafficked back in fairly quickly. In addition, we're now aware that some membrane microdomains are being formed, and this is very recent, between sphingolipids and specific sterol connections at the plasma membrane. But we don't know the relevance of those yet, so that's kind of exciting. We know that there's increased unsaturation of lipids that happens both at the ER and in the chloroplast, and that's critical. We know that those get trafficked other places, in part by action of SFR2, which my lab's worked on. This takes apart some monogalactolipid, which is heavily prevalent in the chloroplast, stacking the head groups. And those diacylglycerols that result end up going back into the ER and then ultimately getting incorporated into TAG. I won't talk about much of that because I assumed that Phil might, but safe to say that there are a lot of changes that have been studied. There are a lot more that need to be studied. The mitochondria, we know that there are changes there. We can measure those, and people have, but we actually know very little about why and how that preserves mitochondrial function. Okay, so the short summary is that most membranes are remodeled. There's more things that happen. It's not just a lipid world. Soluble sugars and amino acids accumulate in liquid spaces. The cytoplasm is the most obvious. They also accumulate in the stroma. And this helps to actually stabilize the membranes further. It helps to stabilize head group interactions there, but it, of course, also decreases the freezing point. And then there's just a whole host of central metabolic responses, which are very meaningful and help to reduce reactive oxygen species. Reactive oxygen species, again, affect membranes. They damage the lipids because they're soaked up by those double bonds, leading to production of stress hormones. And so there's some meat here also for lipids. So let's get into some of those lipid changes. Why are the lipid changes that happen? And do we understand the biophysical principles behind them? And the answer is to some extent, and for the classic lipids, we do. So this is the building block model, which is oversimplified, but with that caveat, it's still useful. Essentially, things that interact better are more rigid. Things that interact worse are more fluid. We have some ideas about what makes lipids interact better or worse. And we know which classes of lipids typically fall into these categories. The things that make membrane lipids interact better are typically that they have a similar size and shape and space fill and chemistry of the head group that matches their tails, allowing them to stack nicely and leave very little space in between each other. The more space that water is allowed to enter the membrane, the more disruptive it is. And that's exactly what membrane fluidizing lipids do. These are shaped irregularly, either because they have very small head groups compared to their fatty acids, or because they are in fact shorter than the rest of the lipids, as is the case for sterols, or because they have been cut like lysolipids and they have half of the fatty acid they used to have. Of course, it's a little bit more complex than only a head group story because the fatty acid chains are also changing. The space taken up by a unsaturated lipid compared to, a fully saturated lipid compared to an unsaturated lipid is importantly different. And when we get to sphingolipids, there are additional deviations from a saturated state that can occur. So when we measure them, do we see things that are similar? So this is one part of one of multiple tables from the review. And this is their best case scenario. This is when we look at similar temperatures, in other words, all freezing, all damaging temperatures in the same variety of Arabidopsis. And then we can find some similar lipid changes across different labs and different studies. Specifically, what we see are that non-bilayer forming lipids like diacylglycerol, lysolipids that are likely important for signaling and likely the result of unknown lipases, phosphatidic acid, as already mentioned, go up. Triacylglycerol, storage lipid, goes up. And we also see reduction of monogalactolipid, possibly because it is being restructured into other lipids. Everything else is less clear. And as soon as we start switching species, there are not a lot of similarities. Part of what my lab has been trying to ask is why. One of the potential answers comes from rhythmicity. It's not just cold consistently. Cold changes between the middle of the day and the middle of the night. And this idea has a lot of weight in terms of transcriptional control of cold. So this comes from this study. But it's a very simple idea, which is that if you have a signal which is stronger at one time of day than the other time of day, and an impetus like cold, you'll document changes where the change is consistent, but you'll miss changes of phase and amplitude because they'll just look variable. And that's unfortunate. And so we measured if that was the case in Arabidopsis and multiple other species, and it clearly is. And I'll just point out for digalactolipid, so this was the same one that we saw variants in some of the Columbia studies. We also in Columbia showed that its phase changed in response to cold. That might be an explanation. During that study, we got asked often, is it the fact that those plants are so different from each other? So we tested a whole bunch of sorghum varieties, all genetically quite similar, Fangyi, who is a recently graduated PhD student and looking for a job, please let me know if you have one. She's fabulous. Found 2 quite tolerant varieties, compared them to a sensitive variety. Here you're looking at the amount of cells broken versus cells intact, mostly broken in the sensitive variety, more intact in the tolerant varieties, and then looked at all the lipids that had changed. And what she showed is that there are very few similarities. This is looking at those log fold changes of these lipids up close. And in a variety of conditions in those tolerant varieties, they look similar. But a lot of other lipid changes were different. We don't know why. We don't know a lot of things. Here's a list of all the things we don't know. But I think everybody's going to enjoy adding to this. We don't actually understand what the role of hexagonal 2 phases are, or those plasma nanodomains. We don't understand, even though phytosterols are everywhere, we only understand their use in the plasma membrane. And even that, only a little. In cold, we also accumulate unusual lipids, but we actually don't understand the biophysics of how these interact in a membrane environment. We don't understand what happens to organelle membranes in the cold. The most studies have been done on the chloroplast, but it keeps going. So with that, I'm going to say thank you very much to everybody who worked on this project with me, and invite you to join us at the Midwest ASPB conference in March. Thank you very much for your attention.
Thank you for the interesting talk, Rebecca. I think we already have a few questions. And the first question is, I think, related to your tag. It says that multiple studies shows that tag accumulations during cold temperatures. Can you outline some of the very specific roles of tag accumulation in cold tolerance related to the cold tolerance?
Yeah, sure. We recently published a paper on this. The theory is that it would help protect the membranes by providing a repository for extra fatty acids that were not perhaps as useful at this time. So we tested a variety of cold conditions and looked at membrane damage and whether or not they were continuous. Whenever you saw lower tag levels, you saw increased damage. We looked at all the mutants that can make tag for this purpose as well. And the answer was that they're not consistent. Sometimes yes, and sometimes no. So we don't think that's the only reason. There's 2 other reasons that have been hypothesized that I have not tested and are still very reasonable, which is that tag is also a reservoir for antioxidants. So in fatty acid breakdown, you can make a lot of NADH. And that NADH may be used or stored in the cell as tag in order to help, not prevent, but restore damage from ROS signaling. And then, I mean, there's more, but yeah.
Well, there are many questions. You can answer them in the chat box, but I think Dave is asking a questions like, first, were the lipid changes also inconsistent in the other Arabidopsis natural lines? Was there any similar pattern in annual, perennial, or overwintering plants? Do you see any changes like lipid?
So there are, great question. We've been trying to answer that exact question. The lipid changes were inconsistent with other Arabidopsis naturally existing lines. Some of the questions behind that are, those other natural existing lines presumably require slightly different temperatures to acclimate to the same level as Columbia. Are the differences that we're seeing related to their different acclimation levels and therefore different amounts of stress? Or are the changes that we're seeing indicative of different responses? We were hoping to ask that with the sorghum study, but as you saw, there were a lot of differences, more so than we expected, and very few similarities. In terms of annual, perennial, and overwintering plants, again, the only ways those have been tested is head-to-head. But we know that those plants require different cold treatments for the same effect. Overwintering plants require a lot more cold, a lot longer length of cold to overwinter. So they're set up with different responses.
Very quickly, I'm adding to this, like Nishant is asking which sorghum variety you included in the study and how it impacts, does it impacts overall or something?
So we started with about 100 accessions in the variety, and we narrowed down to 10. The 3 I showed are BTX 623 for the sensitive line, Cao Ling for the tolerant line, one of the 2 tolerant lines, and HKZ, which I'm sorry, I'd have to look up that abbreviation for the other tolerant line, but I can do that in the chat.
Okay, thank you, Rebecca. It's time to move our second speaker, Yingqi Cai. She is a research assistant professor at University of North Texas, where she earned her PhD degree in biochemistry and molecular biology. After doctoral degree, she joined Brookhaven National Laboratory for her postdoc, where she investigated the regulatory mechanism of plant lipid metabolism and developed strategies to enhance lipid production in plant vegetative tissues without compromising the plant growth. Her research interests including plant lipid metabolism, storage lipid compartmentations, and metabolic engineering in plants. Her current research focuses on understanding the cellular mechanism underlying the biogenesis and modulation of lipid droplets, subcellular organelles that stores neutral lipids in the aqueous cytoplasm and play an important role in plant development, stress response, and the energy storage. Today, she will tell us about lipid droplet biogenesis in plant cells. Yingqi, the stage is yours now. Thank you.
Okay, let me share my slides. All right. Thank you, Maneesh, for the introduction, and thanks to the editors and the organizing committee for this opportunity to share my research here. Well, today I will talk about plant lipids from a cell biology perspective, mainly focusing on how vegetable oils are packaged into lipid droplets. So due to time limitation, I will highlight only a few studies. Please check out our review article for more detailed and comprehensive overview of plant lipid droplets. Okay, now let's talk about lipid droplets. So lipid droplets are cell organelles used for lipid storage. They consist of a hydrophobic core of neutral lipids, usually triacylglycerols, surrounded by a monolayer of phospholipids, with some proteins embedded on the surface of lipid droplets. This way, the hydrophobic neutral lipids can be stably stored in the aqueous cell environment. In plants, lipid droplets are usually produced accumulated in seed tissues, where they are used to store energy and carbon. During seed germination, these lipid droplets can be degraded to provide carbon, energy, and membranes for seedling establishment. It was recently noticed that while in low abundance, lipid droplets also exist in plant vegetative tissues, and they are involved in many biological processes in vegetative tissues. Despite the importance of lipid droplets, we didn't know much about how they are formed and regulated in plant systems. We knew that they are derived from the ER, where neutral lipids are synthesized. And a few lipid droplet-related proteins have been identified in animal systems. However, we didn't know the conservation level of this process in plant cells. So to learn more about lipid droplets in plants, a necessary step was to find more lipid droplet-related proteins in plants. So today I will start by talking about the 3 primary strategies used to identify novel lipid droplet proteins in plants. Then I'll briefly share some studies that explore the functional mechanisms of some lipid droplet proteins. And finally, I'll conclude with a brief overview of future directions in lipid droplet formation, in lipid droplet research in plants. Okay, now let's start with the identification of novel lipid droplet-related proteins in plants. So the first strategy that we took was homology-based searches for plant homologs of animal lipid droplet-related proteins. So basically, we know that some proteins are important for fat storage in human and animals. So we looked for their homologs in plants using based on their sequence similarities and conserved functional domains. Then we tested the functions of these plant homologs in lipid droplet formation in plant cells. Okay, that's the workflow of our first strategy. Using this strategy, one important protein was found in plant systems is CPIN. So this protein was firstly identified as the causal protein in a human lipodystrophy disease. Patients with this disease will have disrupted lipid storage. Like you can see here, they lose fat in adipose tissues and accumulate fat in tissues that are not supposed to store fat, like muscle and liver tissues. And disruption of CPIN in yeast cells also cause lipid packaging defects, showing fewer and larger lipid droplets in the CPIN mutant cells compared to the wild type. It's believed that the CPIN proteins can form a complex at the ER lipid droplet junction site, where they function as a vent to facilitate the partitioning of neutral lipids from the ER into lipid droplets. So when we looked into plant genomes, we found that unlike the single CPIN isoform found in human and yeast, vascular plants usually have multiple CPIN isoforms. For example, in Arabidopsis, there are 3 CPIN isoforms. And we found that all 3 Arabidopsis CPINs can promote lipid droplet formation in plant cells in a manner similar to that in human and yeast cells. However, CPIN1 produces more large-sized lipid droplets, while CPIN2 and especially CPIN3 produce more smaller-sized lipid droplets, suggesting a plant-specific functional mechanism of CPIN in lipid droplet formation. Okay, well, these homology-based searches have been very effective, and this CPIN is just one example of the proteins that we have identified using this method. So we realized that plants may have some unique lipid droplet proteins that do not exist in animal systems. So how do we find them? One approach we took is lipid droplet proteomic analysis. So basically, we extract the lipid droplets from plant tissues and extract the proteins from those isolated lipid droplets, and then use mass spec sequencing to identify those lipid droplet-associated proteins. One important protein identified using this method is called lipid droplet-associated protein, LDAP. So this protein was firstly identified by Dr. Patrick Horn, who's also the co-author of this review paper. He found this protein from lipid droplets in avocado mesocarp, which is a vegetative tissue accumulating oils. Okay, and then Dr. Robert Mullen's group characterized the Arabidopsis homolog of this protein, and he found that this LDAP protein localizes specifically to the surface of lipid droplets and is involved in the dynamic regulation of lipid droplets in plant vegetative tissues during diurnal cycles and under stress conditions. Okay, so that's the second strategy. With proteins identified using these 2 strategies, we further found more related proteins by interaction-based studies using yeast-to-hybrid screening and affinity capture assays. So in our affinity capture assay, we used the transient expression system in benzamine ANA to pull down GFP-tagged lipid droplet proteins together with their interactors. Since leaf tissues don't normally produce much storage lipids, we use a seed-specific transcription factor called LAC2 to promote lipid production and to simulate a seed environment in leaf tissue so that we can pull down some seed-specific lipid droplet proteins. Okay, these interaction-based assays have not only uncovered interactions between non-lipid droplet proteins, but also identified more new lipid droplet proteins that had never been associated with lipid droplets before. For example, a protein with unknown function was found interacting with LDAP on the surface of lipid droplets. So they named this protein LDAP interacting protein, LDIP. Interestingly, I found this protein also interacting with the SAPN and the ER. So the short summary of this story is that the LD protein play an important role in different stages of lipid droplet biogenesis and the modulation of lipid droplets. All right, so given that these efforts have greatly expanded the inventory of lipid droplet proteins in plants, our next step was to look at the structural features and functional mechanisms of these plant lipid droplet proteins. Now I'll share just a few of our studies as an example. Remember that I mentioned different Arabidopsis SAPN isoforms produce different size lipid droplets. So we asked what structural features that confer the specialized functions of different SAPN isoforms in plant cells. Here shows the topology model of the 3 Arabidopsis SAPNs. You can see that all 3 SAPNs consist of 2 conserved transmembrane domains, flanking a ER luminal region. Compared to SAPN 1, SAPN 2 and SAPN 3 has a relatively longer N terminus before the first transmembrane domain. And to test whether this N terminus is responsible for making different size lipid droplets, we swapped the N-terminal region between SAPN 1 and SAPN 3 and expressed this chimera protein in Basamiana leaf tissue and looked at the lipid droplet phenotype. Here you can see that the protein with the SAPN 1 N-terminus and the SAPN 3 C-terminus produced large lipid droplets similar to SAPN 1. And the protein with SAPN 3 N-terminus and SAPN 1 C-terminus produce smaller size lipid droplets similar to SAPN 3. Meaning that the N-terminal region of SAPN is important for determining the size of lipid droplets. Some homology modeling of SAPN structures revealed a hydrophobic helix in the ER luminal region of SAPN, which can insert into the ER membrane and is both necessary and essential for interacting with LDIP. In LDIP, there is a strongly hydrophobic segment that can insert into the ER membrane and interact with SAPN. And we found that disruption of either SAPN or LDIP can cause the accumulation of aberrant supersized lipid droplets in Arabidopsis C tissue. So that means the interaction between SAPN and LDIP is necessary for appropriate formation of lipid droplets in plant cells. And this interaction may happen at the very early stages of lipid droplet formation. Okay, significant progress has been made in the past few years to understand lipid droplets in plant cells. Questions still remain to be addressed. So now let's take a quick look at some future directions of lipid droplet research. The first question is, how do proteins localize to lipid droplets? We know that some lipid droplet proteins have a hydrophobic domain that can insert into the hydrophobic core of lipid droplets, while many others only have a amphipathic domain that can bind to the phospholipid monolayer of lipid droplets. In either scenario, the mechanisms by which these proteins recognize and localize to lipid droplets remain unclear. So future structural analysis of lipid droplet proteins and molecular simulation of protein-lipid interactions will likely provide valuable insights into this question. Well, plant lipid droplets have been getting more attention lately because they are found to be involved in many different biological processes. So one of the next big areas of lipid droplet research will be looking at how lipid droplets participate in different plant physiological processes. We know that lipid droplets from different tissues are covered by proteins, by different types of proteins. Some of these proteins have enzyme activities, and some of these proteins are related to stress conditions. By exploring the functions of these lipid droplet proteins, we can better understand how lipid droplets are functioning in all sorts of biological processes. Okay, with all that, I would like to thank my co-author, Dr. Patrick Horn, for his contribution to this review paper. And a big thank you to our plant lipid community for advancing our knowledge in lipid droplet biology in plants. And the studies I presented today are the result of many years of hard work from many students and postdocs from many groups. So I apologize that I don't have the space to list all of them here. And finally, I would like to thank the DOE and the University of North Texas for funding our research. All right, thank you for listening. Now I can take any questions that you may have.
Thank you, Yingqi. There's a time for only one question, and Usman is asking, are there trade-offs associated with increasing lipid droplet accumulation in plant, such as growth retardation or yield reduction? Did you see that?
Can you repeat that question?
Are there any trade-offs associated with increasing lipid droplet accumulation in plants, such as growth retardation or yield reduction?
Yes, accumulation of triacylglycerols and lipid droplets is associated with some growth defects. That only happens because usually in leaf tissues, lipid droplets are regulated dynamically and very quickly. So it's normally you don't have a lot of triacylglycerols or lipid droplets accumulated in vegetative tissues. But when we engineer them in vegetative tissues by using some seed-specific enzymes and transcription factors, you will see some defects in plant growth and development.
Okay. Thank you, Yingqi. It's time to move our next and final speaker, Philip Bates. Phil is an associate professor at Institute of Biological Chemistry at Washington State University. His focus is on the elucidation of plant lipids metabolic pathway leading to the production of essential membrane lipids and valuable plant oils, including characterizing how different plants can produce oil with widely different fatty acid compositions and thus have distinct physical and chemical properties. One key approach his lab is taking is tracing carbon fluxes through the lipid metabolic network, utilizing isotopic labeling to discover novel metabolic pathways or identify metabolic adaptations due to mutations or genetic engineering. Today, he will share his understanding of lipid metabolic dynamics for pathway discovery and identifying bottlenecks within bioengineering. Thank you, and over to you, Phil.
Great, thank you. And I'd again like to thank the organizers for inviting me. Today, I'm going to focus on talking about one story from my lab, which is a nice follow-up from the question for Yingqi, which talks about what happens when you accumulate oil bodies in plants. And so we'll just get started onto that. Okay, so everyone knows plant oils are an important part of our daily lives. We grow lots of plants for the oils. These include canola, soybeans, corn oil is part of it, but not the main product. Sunflower seeds are used to make oil. And most of this goes into food or feed or cooking applications. But plant oils can also be used for making plastics, various different paints and polymers, soaps and resins. And in particular, biofuels such as biodiesel or sustainable aviation fuel or renewable diesel. So what gives plant oils these properties, these useful properties, has to do with the fatty acid composition of the triacylglycerol molecules that make up plant oils. One key part, if we focus on a energy or fuel application, is the fact that fatty acids are highly reduced hydrocarbon structures, and this gives them inherent high energy content compared to things that are more oxidized, such as carbohydrates. And so there's more than twice the energy content per gram in fatty acids than in sugars. And this is important if we want to use them for biofuels. As I just mentioned, triacylglycerols can be used as a drop-in replacement for various different biofuels after some refining for those purposes. And so as we move towards wanting to replace petroleum for various different reasons, including pollution and climate change and other sorts of things, plants can contribute to this because plants take up carbon dioxide, and then when you use them as a fuel, it releases it. And so you can, in theory, get to sort of close to a net zero emissions. So if we want to use more plant oils for fuels and things like that, we also have to consider the effect on food. And with the rising human population means we need more food, more fuels, and more chemicals. And if plant oils are going to contribute to that, we have this food versus fuel debate. And so we don't want to just take our food oils and convert them into using chemicals and biofuels. We need sort of alternative crops. The other thing we have to think of is we can't just replace our agricultural crops because agricultural land is limited. And as Rebecca nicely indicated, that we can only grow certain times of the year because of cold, like we have going on right now. The other thing is when you think about for most of our oil crops, such as soybeans shown here, 2/3 of their life cycle, they're building the vegetative structures to capture carbon and photosynthesis. And that is not going to produce oils. Only in the last third of the life cycle do they develop the seeds that ultimately lead to the accumulation of the oils. And so what we really need to do is we need to enhance total productivity per acre of land and be able to utilize more of that time to accumulate oils. And so one way we could do that is having a vegetative oil crop that is a large biomass crop, something that produces a lot of leafy material like switchgrass or maybe tobacco, and that if it can accumulate oil through its entire life cycle, that you can then in theory get more oil per acre of land than you can from an oilseed crop. And so I like to call these obese plants because they're accumulating fat throughout their entire vegetative tissues. So can we do this? And the answer is yes. It's been demonstrated quite a few times, but getting a lot so that we can really have high yields is the tricky part. So the general approach is what's been called a push-pull-protect approach, whereas fatty acids are made from sugars. And if you upregulate transcription factors such as wrinkled 1, it enhances glycolysis and fatty acid biosynthesis. And so this is the push to make more fatty acids. Then it turns out in leaves, almost all the enzymatic steps to make triacylglycerol are already there because they're also used to make membrane lipids. So the substrate to make triacylglycerol is diacylglycerol, and you just have to add one more fatty acid to make triacylglycerol. And the key enzyme for that is a diacylglycerol acyltransferase, or DGAT. And so if you also upregulate DGATs, you have a pull of the fatty acids into triacylglycerols instead of into membrane lipids. So you have a push and a pull. And then, as Yingqi nicely mentioned, triacylglycerols can be unstable. And so if you want to accumulate a lot of them, you need to stabilize their accumulation. And so this is the protect portion. And this can be done by adding oil body proteins that limit its degradation, such as oleosin, and other approaches can limit tag turnover and beta oxidation. And so this has been done a bunch of times and in various different plants, bioenergy type plants like sorghum and sugarcane. Duckweed is a newer model. And most of the time you get less than 10% of the dry weight as triacylglycerols. However, what has worked the best is in tobacco. So my people here, so they're not in my way of my cursor. CSIRO in Australia has developed multiple different lines of tobacco that has seed oil-like yields of oils in leafy tissue. The first generation used this push-pull protect with wrinkled 1, DGAT, and oleosin, and it produced what they call the high oil line, or HO, that accumulated up to 15% oil by weight in the leaf. And for those who don't know, normally tag in leaves is less than 1%, a lot of times less, more like 0.1%. And what happened, as Yingqi briefly said after the question, was that we had oil accumulation. However, it led to reduced growth. And this, the flowering stage doesn't look so bad. At earlier vegetative stages, the reduction in growth is quite large. And so one thought is, well, you're moving carbon from what's available for sugars and into TAG, and so it just doesn't have enough for growth. And so that's a possibility. To try to enhance the oil content even more, they added another transcription factor, LEC2, and to boost the oil content, but it had a surprising effect. Oil content doubled, but growth recovered, almost to wild type. So that's telling us that it's not just the shift of carbon from sugars for growth to oil, but there's some sort of change in metabolism happening. So this is when I came into the project. Colleagues at CSIRO contacted me, and because my lab studies dynamics of metabolism, particularly in lipid metabolism, they asked if we could take a look at this. And so this is the story I'm going to talk about today. So if you think about what kind of changes could happen in metabolism that might affect growth, you have to think about how metabolism is regulated and ways to do that. And it turns out that there's many ways to regulate metabolism. Things like mRNA abundance, protein abundance, post-translational modifications, protein-protein interactions, small molecule regulators of individual enzymes, where you can have substrate activation or product inhibition. Enzymes can be deactivated, and there's multiple ways, right? And so some approaches may take some omics approaches like transcriptomics, and you look at one of these things, but you typically won't look at them all together. And prior transcriptomics on the HO line didn't have any smoking guns on why the growth was reduced. And so we took a metabolic flux approach. And for those who aren't familiar, metabolic flux is essentially tracking the movement of carbon-based molecules through metabolism from precursors to products. And this involves the sum of all forms of regulation, because we're following the actual carbon moving through metabolism. And if we find bottlenecks or changes in metabolic flux, we can then go back and look at what forms of metabolic regulation affected this. Okay, so the approach we took is we started with CO2 that was carbon-14 labeled, and we fed it to the plant, and it's going to go through all metabolism, including lipid metabolism here. And we used the pulse chase type approach, where this is where we pulse with CO2 for 2 hours, and then we remove the 14C-CO2 and we let them grow normally. And then what we're seeing is that at early time points are the things that are initially made, and they get labeled. And then over time, those initial products will turn into other things as it moves through metabolism. And we can learn about precursor product relationships. Okay, so the setup of this experiment was we had a large chamber that we put in all of our tobacco lines in replicates, and we pulsed for 2 hours. And then we let them grow normally. And then over 145 hours, we sampled by taking leaf discs. And you can see this is what the plants look like as going through the experiment. Tobacco is actually quite robust. You can take discs out and it doesn't affect their growth hardly at all. And then what we did is we fractionated this into different compounds. So we had major fractions of the lipid soluble fractions, aqueous soluble small molecules, protein fraction, starch fraction, insoluble cell wall. And then we divided the lipid soluble fraction into triacylglycerols or oils, the polar membrane lipids, and a few other compounds. Then also the aqueous soluble compounds into amino acids, organic acids, and sugars. I won't be able to talk about all these today for limited time, but I'm going to talk about some of the highlights. This graph down here at the bottom just shows the accumulation of total mass of lipids, not 14C, over time. And you can see the LEC2 line accumulates a lot. The HO line has more than wild type at the start, but it stays roughly constant the amount of lipid, which is what we measured a few times. And then the wild type grows quite a bit, and it sort of catches up later on. But this is mostly membrane lipids, as you will see. Okay, so key points I'm going to focus on from our data. One is just after we fractionated out, we added up all the carbon-14 that was captured. And one of the most interesting things we found right away is that this first time point is that wild type had the most carbon capture, and that the reduced in stature high initial first generation high oil line had about half as much carbon capture. And so this is one of the first things that we didn't expect, but might lead to why the plants were reducing growth, is they're just not capturing as much carbon. Interestingly, when the LEC2 transcription factor, which was to boost oil accumulation, was also expressed, the carbon capture increases. And so the changes in metabolism didn't only affect lipid metabolism, but they affected overall photosynthesis. The general decrease across the time course that you see here is because leaves are a source tissue, and then most are a large part of that gets transported throughout the plant through the phloem to other sink tissues. Other big parts that we saw big changes was cell wall components in the initial HO line, despite its reduced stature, had a lot more carbon flux into cell walls. And then we had interesting changes in starch and lipids that I'll get into in a little bit more details. Okay, so what we're showing here is how the carbon flux through metabolism into lipids. The column on the left is polar lipids, and this is total radioactivity, percent of the total, and then percent of the total lipids. And the general feature for the polar lipids or the membrane lipids is that they're relatively constant over the time course, which makes sense. Membrane lipids don't turn over very much, but that wild type had more, and that the oil accumulating lines had a bit less, and that was somewhat expected. What we didn't expect was the dynamics of the oil between the lines. So the wild type in black has very little oil in the leaves. The first generation oil line, you can see that it goes up and then it comes down, kind of goes up again, and then over the prolonged time course mostly goes away, even though the mass of the oil stays constant. So what this is telling us is that there is both synthesis and turnover going on at the same time. Interestingly, the LAC2 line that had the enhanced oil accumulation was a lot more constant in its accumulation of oil. And so that was a key effect of what we think is happening in these lines. The other thing is we wanted to compare the oil and other compounds. So I'll remind you, this is a pulse chase experiment, which means that at the time zero is when we remove the radio label. So anything that's being synthesized after this is coming from something else that's already been synthesized in the plant. And so what you can see is over the first day, triacylglycerol increases in red, and blue is starch. And this is the high oil line. And then at night, starch goes down, which is sort of well characterized. Plants use starch to feed metabolism during night. But as starch goes down, oil continues to go up, which suggests that carbon is moving from starch to oils. But what was really interesting is during the next day, the oil decreased and starch went back up, right? And so this suggests that carbon is moving from fats to starch. And you can kind of see that this accumulates then during the next night, as we have the same feature, is that tag goes up as starch goes down, and it kind of starts to, as the carbon gets diluted out of the leaf, peter out over the next few days. However, in the line with a lot more oil accumulation, we have a lot more constant accumulation of oil. So the key thing here is that there's dynamic diurnal and inverse cycling of carbon in the line with reduced stature, and that the line with the higher oil content and better growth, the oil is a lot more stable. And so what does this mean towards metabolism? So what we think is going on is that in the high oil line, you capture CO2, and this goes through glycolysis and fatty acid biosynthesis in the tag. However, that tag is not stable, and it turns over and goes through beta oxidation. That carbon ends up in starch. And the only way to do that plants can do and that animals cannot, is through what's called the glyoxylate cycle. And the glyoxylate cycle takes place in the peroxisome, which is here in blue. The other thing that takes place in the peroxisome that is really important for photosynthesis is photorespiration. And they both have glyoxylate as an intermediate. And when we think it's happening is we're having overproduction of glyoxylate through the beta oxidation. And why is that important? Well, glyoxylate is known to be a key inhibitor of multiple steps of photosynthesis, including rubisco and rubisco activase. It's also known to be inhibitors of photorespiration, which is essential for overall photosynthesis. And so what we think is happening is that when the tag is not stable, it inhibits overall photosynthesis by having the glyoxylate cycle running in a leaf, which normally does not run in a leaf. It only runs in a germinating oilseed. And so we're having metabolism occurring where it shouldn't be. However, if you can stabilize that tag, as in the LAC2 line, you no longer have beta oxidation going on or much reduced. It doesn't affect glyoxylate levels, and then that would alleviate this reduction in photosynthesis and photorespiration and allow for enhanced growth and enhanced carbon uptake. And so That's the brief overview of our most recent publication. And I want to mention just a few key points, which is that metabolic engineering of obese crops can produce unexpected changes in metabolism. And that flux analysis can reveal unexpected dynamics that are not clear from mass abundance or transcript levels alone. And that tag stability seems to be key to both high oil accumulation and limiting adverse effects on metabolism and plant growth in these obese crops. And then considering that the LEC2 line still did not have wild type CO2 uptake, further increases in CO2 uptake in the LEC2 line may actually lead to higher leaf oil, and that should be a new direction of engineering. And with that, I'm happy to take any questions.
Thank you for a great talk, Phil. There are time for only 2 questions, although there are multiple. But the first question is from Emma. Have different groups looked at engineering tag lipid accumulation in other tissue types other than leaves?
Yeah, that's a great question. So there was a paper on potatoes. I think they made up to, I didn't list all of them on my little brief overview slide. And of course, in potato tubers, I think they got 3 or 4%. And so yes, tag's been tried to accumulate in various other tissues. It's tricky because we still don't fully understand all the steps that are involved in even triacylglycerol biosynthesis in different tissues and in different plants. Other research in my lab goes to that. And as Yingqi indicated, we still don't understand all the steps involved in accurately packaging the oil. And so I think to really get a engineered biomass crop that accumulates a lot of triacylglycerols in various different vegetative tissues, there's a lot more basic science we need to learn.
Okay. And the second question is from Essa. How the increase in particular fatty acid concentration was reflected in any of the lipid class, or it's a whole profile?
Oh, that's a great question. So our prior paper to this one, also in Plant Physiology in 2020, we demonstrate that in the high oil line, there was reduced flux of fatty acid biosynthesis into membrane lipids, particularly into PC and galactolipids. And so that we believe what's happening is that there's competition for substrate, particularly diacylglycerol, for producing membrane lipids versus triacylglycerol. Interestingly, in this current publication, we show that the LEC2 line that has twice as much triacylglycerol, there's no further reduction in membrane lipids. And so it seems that the plants can handle a little bit of reduction in membrane lipids, but you can accumulate more oil without having further reductions. And I think that a lot of that has to do with the stability of the oil is the key thing that's limiting it, not so much the biosynthesis.
Okay, I think with that, I will thank all the speakers, Mary, and all the attendees, whoever joined us for today for the webinar. Thank you so much, and bye-bye.
Bye-bye.
Thank you, everyone. And keep an eye on our next webinars. I think we have one in May for Plant Cell on translating from Arabidopsis into other systems. So thank you very much. Bye-bye. Thanks for coming.
Thank you very much.